Hi, it's Dr. Isom, and I'm back to give you the third part of the Chapter 2 Research Methods lecture. This one is on personality research design. I'm going to talk about 3 different research designs that are commonly used in personality research, and that is the case study, the experiment, and correlational studies. The case study is the first one I want to talk about, and that is when you simply do a very detailed, in-depth examination of a person, including personality measurement, behavioral observations, informant reports, any source of data that you choose to use. It can be quantitative, it could be qualitative, it could include long interviews with that individual. But the whole point is that you want to have a really detailed explanation of the individual, their behaviors, and the potential reasons for why they engage in the behaviors that they do. One of the things that can result from the case study is the development of general hypotheses and scientific principles that may be used in other research designs to further investigate. 2 very famous psychologists who use the case study method are Freud and Piaget. Freud based his psychosexual theories of development and his psychoanalytic theory in general on case studies of his clients. Another famous psychologist who used the case study method to develop general principles is Piaget. Piaget is well known for his developmental theories of cognitive ability, and he actually based those theories on case studies of his children. He studied his children to determine at what age specific cognitive abilities appeared, and then he created general principles and then a theory of cognitive development based on his in-depth examination of his children. Case studies are great because if you really want to get a very detailed description of the whole person in their natural environment with the highest precision possible, then the case study is the method that you should use. Like I said, it can be a source for ideas for generating hypotheses about why individuals behave in the ways that they do. And sometimes it's necessary because it's the only way you can fully understand a person. It comes with its disadvantages, though. The most obvious one is low generalizability. What may be true for one person in terms of what motivates their behavior may be completely different for other people. And so any principles that you identify in a case study need to be tested using other research methods. The other thing is you don't have any control over the variables. You can't manipulate variables to see how the person will respond. And any general principles that you are able to identify in a case study need to be tested using other research methods. And sometimes that can be a difficult thing to do. But this didn't stop Piaget or Freud from developing their very famous theories based on case studies. Experiments are the next research design. In an experiment, you're comparing how different groups respond to different levels of an independent variable. The idea is that you're wanting to determine whether changes in an independent variable or different levels of an independent variable cause a change in your dependent variable. There's 2 really important things that have to be present in an experiment, and that is you need to be able to manipulate your independent variable. The variable that you think is causing an effect on the dependent variable has to be controlled, and it has to be manipulated so that different experimental groups get different levels of that variable. The second thing that needs to be there is that your different experimental groups, the ones that get different levels of the independent variable, need to be as similar as possible. They need to be randomly assigned or randomly selected from your sample so that the groups are statistically as similar as possible. The benefit of using an experiment as your research design is that if it's done correctly, it allows the researcher to make a claim about one variable, the independent variable, causing a change in the dependent variable. So it allows you to determine causality, whether one variable causes another to change. Okay, what do you have to do in an experiment to be able to show causality, to show that the independent variable caused a change in the dependent variable? Well, the first thing, as I mentioned before, you need to make sure that your groups are essentially equivalent to each other. One of the ways to do this is to randomly assign or randomly select participants into each of the experimental groups to make them as equivalent as possible, and so that participants only get assigned to any particular group due to chance. What this does is has the impact of making the groups as equivalent as possible because any individual differences for any of the participants will be randomly assigned into the groups so that they should balance each other out. The second thing that has to be there is control of the independent variable so that each group gets a different level of the independent variable as a treatment so that you can compare the response of each of the groups for different levels of that independent variable. Once your treatment groups are exposed to different levels of your independent variable, you can measure the dependent variable for each of the different treatment groups and compare whether the independent variable caused a difference in the level of the dependent variable between groups. There will be some differences, small differences between groups, just because of chance. But if you observe a large difference between treatment groups, then you can do a statistical test to determine whether those differences are greater than what you would expect by chance. It might help to give you an example of an experiment. And this experiment example is based on research several years ago that suggested that consuming berries and other high antioxidant fruit may have a positive impact on cognitive abilities. So the research question that we'll investigate with this experiment is whether blueberries or consumption of blueberries increases a person's short-term memory ability. We can identify the independent variable and the dependent variable, and then I'll talk about the procedure that needs to happen for this to be an experiment. First, let's identify the independent variable. It is blueberries, because the question is whether blueberries cause an increase in short-term memory abilities. Next, we'll identify the dependent variable. The dependent variable is the variable that you're expecting to change as a result of the independent variable. The dependent variable in this case would be short-term memory ability. For the independent variable, we'll take a 4-week period and have participants consume different amounts of blueberries over that 4-week period to see whether it has an impact on their short-term memory abilities. We'll measure short-term memory abilities at the beginning of that 4-week period and then again at the end of the 4-week period by using a common test of short-term memory, and that is a digit span test. And this is simply when the researcher or the experimenter lists a series of up to 12 to 15 digits and then simply has the participant repeat back all the digits that he or she can remember. The number of digits that's remembered can be considered a measure of that person's short-term memory ability. The next thing to do in developing this experiment is to create your treatment groups. First, we'll randomly assign participants into 3 different groups. In that way, we are assuring, as much as possible, that those groups are roughly equivalent to each other. And then we'll assign each group a different amount of blueberries to consume over that 4-week period so that we can assess whether consumption of blueberries has an impact on short-term memory. So the first group will be the no blueberries group, and that's essentially your control group. Okay, they are not getting the treatment. The second group, we'll assign them half a cup a day of blueberries to eat. And then the third group, we'll have them eat a cup of blueberries per day. So each of our groups for 4 weeks will eat the amount of blueberries that corresponds with which treatment group that they're in. After the 4-week period, we'll bring them back in and measure each participant's short-term memory. What we have here is we have an independent variable that we are manipulating into different levels, and we have groups that are randomly assigned to increase our ability to control that the groups will not be different for any reason other than the amount of blueberries that they consume. Once we've measured all of the participants' short-term memory, then we'll average the short-term memory test scores for people within each group and compare the short-term memory scores for each of the groups. If we see differences between the groups, then we can use a statistical test to see if the groups are significantly different. In other words, that they are showing differences that you would not expect due to chance. So I've gone ahead and plotted the data here. What this data is demonstrating is the average number of digits recalled for each of the 3 experimental groups. So the first one is the control group that consumed no blueberries. You can see that they, on average, remembered 4 digits in the digit span test. The second group is the group that had half a cup a day, and you can see that on average that they recalled about roughly 5 digits in the short-term memory test. The group that showed the best short-term memory is the 1 cup a day blueberry group, and you can see that they recalled on average 8 digits. Now this difference between this experimental group and the other 2 experimental groups may be due to the amount of blueberries. It may be due to the independent variable, and that's something that you can test with the t-test or with an ANOVA. And if you determine that this group that consumed 1 cup a day of blueberries remembered significantly more digits than the other 2 groups, then the researcher could say eating blueberries had a significant impact on short-term memory ability, at least as measured by the digit span test. So those are the basics about the experimental method. Sometimes there's some issues that might arise with experiments, and one of the important ones is that sometimes there might be some uncertainty as to whether any effects that you have observed in an experiment are really due to levels of the independent variable. For example, in this study, it could be that the group that was eating a cup of blueberries a day, which seems like a pretty significant amount, started not only to eat those blueberries every day, but also thinking about the other types of food choices they were making, and perhaps they actually started eating healthier. And maybe eating healthier also motivated them to exercise regularly. We know that regular exercise not only increases neuroplasticity in the brain, and increases the number of connections between neurons in the brain, but it also has been shown to be associated with the development of new neurons or neurogenesis. Now, either of those things might actually increase a person's short-term memory ability. Now, the problem is, how do we know whether that increase in short-term memory ability was due to the blueberries or the other things that might have changed the neuroplasticity, the neurogenesis, when that person either started eating healthier or exercising on a regular basis? So this is one of the things that happens when we're not sure whether any effect that we have attained or any effect that the experiment shows, when we're not sure if the effect on the dependent variable is really due to the independent variable, that there might be other variables that cause the effect, then we have questions about the internal validity of the study. Whether or not the effect that the researcher observed on the dependent variable was really due to the independent variable. Other issues that happen with experiments, not specifically in the blueberry case, but there may be times when, in order to observe an impact on the dependent variable, levels of the independent variable may have to be higher than you would ever actually encounter. This is true for some of the substances that they've identified as carcinogens. Research has shown that there are some substances that are associated with the development of cancer in lab animals, for example, but the truth is, a person would have to encounter such a large amount of that substance to be equivalent with the amount that was demonstrated to cause cancer in lab animals. It would never actually happen in real life that you would encounter that much of that carcinogenic substance. And another thing that sometimes arises with experiments is that sometimes you have to include deception in your study in order to investigate the research question that you have. And there are times when ethically that is okay, but it raises the question as to the generalizability of those findings that involve deception. The last thing that's really an issue with experiments, and this is a major issue, is that you can't study everything with experiments or with the experimental method, because in order to use the experimental method, you have to be able to manipulate the independent variable. And depending on your research question, that isn't always possible. Let's say, for example, that you were interested in determining whether malnutrition in grade school contributes to greater levels of anxiety and depression as an adult. Even though your research question would require that you randomly assign people into groups and then have one group experience malnutrition where the other one doesn't, so that you can observe the impact of malnutrition on increased anxiety, that's not something that you can ethically do.
And nor would you want to.
So there are things that you might want to study that would be unethical to study in an experiment. There are also research questions that you might have that you cannot manipulate, like the impact of bereavement. You can't obviously manipulate whether someone lives or dies and then observe the impact on their loved ones. So then what do you do for those situations where you can't really study it with an experiment? Well, that's where the correlational study comes into play. Correlational studies are easier to conduct because what they are essentially doing is measuring variables, not manipulating, just measuring variables to determine what variables go together. What variables are related and how related are they? The goal of the correlational study is to determine whether there is a relationship between two variables by simply measuring both variables in the same sample of participants. Once you measure the two variables, Then you plot each person's score on those variables into something called a scatter plot, and then you can compute a correlation between those variables. That correlation will tell you whether there is a relationship between the variables that you measured, and if there is, how strong it is and in what direction. You can test statistically whether there is a relationship between those variables using the correlation coefficient. And as I mentioned, the correlation coefficient will give you a numerical measure of the strength and the direction of the relationship between the two variables that you measured. Sometimes there's no relationship between the variables you measured, and in that case, your correlation coefficient would be 0. But sometimes there is. If you do find a correlation between two variables and plot the two variables in a scatter plot, this is what you might see. Correlations range from negative 1.0 to positive 1.0. On the left, right here, you will see a negative 1.0 correlation. And what that means is that there's a perfect negative correlation such that when the level of one variable goes up, the level of the other variable goes down in the exact amount. On the other side, we have the perfect positive correlation. If you have a perfect positive correlation, what that tells you is that as one variable increases, the other variable increases in exactly the same amount. Now, perfect positive and perfect negative correlations are something that we don't see in psychological research and in personality research, and we'll talk about that in a minute. What we usually see is a less perfect correlation, either negative or positive, or in some cases where there's no relationship between variables, we'll see a correlation of 0. So correlations range from negative 1.0 to 1.0, okay, and those are the perfect correlations. But as I mentioned, this doesn't happen in personality and psychological research. Why is that? Why don't we have any perfect correlations? And why is it difficult to observe much stronger correlations in personality research?
In fact, most correlations in personality go
from the 0.30 range to around the 0.50 range. And we would call these moderate correlations. Why is that? Why don't we see stronger correlations in psychological research and in personality research? Well, the answer goes back to classical test theory. Remember, with classical test theory, the idea is that there is no perfect measurement, and even if you've got a really precise measure, you're never going to get the perfectly true score for any individual on any construct, whether it's personality or weight or anything you could possibly measure. With classical test theory, you get an observed score. That's the score that you get on a test, or that's what you see when you step on a scale. You get a number, right? That's your observed score. But we know that that observed score is always a combination of your true score for whatever you're measuring plus some component of error. Error is always going to be there. It's always present. It can either be random error, so it affects some measures some of the time, or it can be systematic error in which you either have an uncalibrated device that you're using to measure or maybe an error in a scoring key. The point is, there's just too much error in psychological measurement to ever have an extremely high degree of precision. It's possible to get a high degree of precision in measurement, but perfect measures and perfect correlations are not something that happens. Here's an example of a correlational study. Let's say you're wondering whether people who are extroverted tend to also be happier. So what you want to do is you want to determine whether there's a relationship between a person's level of happiness and their level of extroversion. All you have to do is measure both of those variables. You can easily measure happiness with a scale like the Satisfaction with Life scale, and that is scored on a range of 1 to 6. And you can easily measure extroversion. There's several scales of extroversion out there. This one looks like your typical Big 5 measure, and it ranges in score from low 50s all the way to 100. Once you measure these two variables in your large sample, then you plot each person's score for each variable. So for example, this person here scored a 3 on the happiness scale, and it looks like roughly a 63 maybe in the extroversion scale. Each one of these dots represents a different person. Once you have all of the data points plotted, then you can compute your line of best fit, which is essentially a measure of the strength and the direction of the relationship between these two variables. what you're getting here is you're getting an estimate of how correlated those variables are. And in this case, the correlation indicates that there's a 0.25 correlation between happiness and extroversion. Now, in actuality, the correlation is a little bit higher than this, and it is, as this one is, in the positive direction. And what that means is that the happier somebody is, the more likely they are to be extroverted. Or you can say it in the other direction. Extroverted people tend to have higher levels of happiness. The drawback of correlational studies is that the thing that you can't do with them, although you can establish the relationship between two variables and whether those variables are positively or negatively related and how strong they're related, you can never identify whether one variable causes the other. That's not something that correlational studies allow you to do. But the one thing that you can do that is a major advantage of correlational studies over experiments is that you can investigate naturally occurring relationships among variables that you can't experimentally manipulate, like going back to the malnutrition and anxiety question. You can measure whether somebody experienced malnutrition as a child, and you can measure their levels of anxiety, and you can correlate those to identify whether there's a relationship between malnutrition as a child and anxiety as an adult. That's not something you could study with the experimental method. Here's an example of a correlational study using the same essential research question as before. We're investigating whether the consumption of blueberries is associated with better short-term memory. So our research question is slightly different. The research question in this case is eating blueberries associated with greater short-term memory ability? Is there an association between eating blueberries and having better short-term memory? The independent and the dependent variables are the same. The independent variable is the amount of blueberries eaten in a 4-week period, just like with the experiment. And the dependent variable is the score of a short-term memory test, the digit span test. Exactly the same. The procedure is what's different. There are no experimental groups. All you do is create a questionnaire that asks about the consumption of blueberries for each person over the last 4 weeks, and then give them that same short-term memory digit span test. Then what you do to investigate whether these two variables are related is you correlate these two variables and determine whether there's a relationship between eating blueberries and having better short-term memory ability. Here's a graph of what we might expect to find if we did a correlational study on blueberries and short-term memory. On the vertical part of the graph, we have the ounces of blueberries on average that a person eats daily. On the bottom, we have a measure of short-term memory. These are the number of digits recalled in that digit span test. After plotting each person's score for the digit span test with the average amount of ounces of blueberries they ate daily, and here's an example. Each data point on this scatter plot represents each person's number of digits recalled in the short-term memory test, plotted with the average ounces of blueberries that they ate on a daily basis over the last 4-week period. So for example, this person here remembered 6 digits in the digit span test, and they reported on average, these are in multiples of 2, that they ate an average of 4 ounces of blueberries each day. Similarly, here's another person who recalled 12 digits in the digit span, and they reported on average that they ate, 2, 4, 6, 8, 10, 12, 14 ounces of blueberries on average each day. After plotting everybody's score for those two variables, how many blueberries on average a person ate each day in ounces, with the number of digits that they recalled in the digit span test, you can compute a correlation. The correlation in this case is 0.78, which is a positive and fairly strong relationship between blueberries eaten and short-term memory ability. What that means essentially is that eating higher levels of blueberries on a daily basis is associated with better short-term memory ability. So essentially, we've studied the same research question as we did with the experiment, but in this case, we studied it using a correlational method, and we found a similar result. Comparing these two kinds of studies, they're very similar in that both can assess the relationship between variables. And not only do both examine the relationship between variables, but the statistics used in each of these designs are mathematically equivalent. The experiment might use a t-test or an ANOVA, whereas the correlational study uses a correlation. it's possible to mathematically convert a t-test into a correlation. So the statistics for each are mathematically interchangeable. So what is the main difference between the experiment and the correlational study? Well, in the experiment, you're actually manipulating your independent variable. And in the correlation, the independent variable is simply measured along with the dependent variable. The main benefit of experiments is that only experiments can allow you to make any claim about causality. That, let's say, eating blueberries actually causes increases in short-term memory. That's not something that a correlational study will allow you to say. The problem in correlational studies, in trying to determine whether one variable is causing another, is that even if you determine that there is a strong relationship between these two variables, eating blueberries and better short-term memory, we're unable to know, based on the correlational method, whether the blueberries are causing greater short-term memory or greater short-term memory is actually causing a person to eat more blueberries. The causal direction is not something that we can establish when you're simply measuring both variables. The other thing that can happen with correlational studies is something called the third variable problem, and you may have learned this as the lurking variable. Let me give you an example of how this works. It's been shown that the more ice cream that is bought and eaten, the more likely a person is to drown. So wait a minute. The correlation between ice cream consumption and drowning is positive. It's positive 0.52, which is a pretty strong correlation. And what that means is that as you eat more ice cream, your chances of drowning go up. Wait a minute, does this mean that eating more ice cream makes you more likely to drown? The correlation between these two seems pretty high. How could that be? Okay, so the thing to remember with correlations is that you cannot establish causality. You're just simply measuring two variables and seeing if those variables appear to be related. Just because you have a significant correlation does not necessarily mean that one causes the other, and really doesn't mean that they are actually directly related at all. All right, so here's the third variable in this case. Drowning rates, higher drowning rates, are highly correlated with more ice cream sales. So if you weren't looking at this third variable down below here, which is the fact that it's summer when they're measuring these two variables, you might think that you have a higher chance of drowning the more ice cream that is sold. But what's really going on is that there's a third variable, and that variable is the summertime or the summer heat. As it gets hotter outside, more people go swimming, and more people swimming leads to higher chances of somebody drowning. Also, as it gets hotter outside, that leads to an increase in ice cream sales and consumption. So the third variable, in this case the summer heat, is the variable that's actually causing drowning and ice cream sales to appear to be correlated. They're not actually correlated, but each is correlated with the fact that the temperature outside has gotten much higher and made it more likely for people to both swim and eat ice cream. The other issue, it's called the directionality problem, is that when you do find a correlation between two variables, you can never really know which variable is causing another in a correlational study. As I mentioned in the case of the blueberries, just because a higher consumption of blueberries is associated with greater short-term memory doesn't mean that eating more blueberries increases your short-term memory. It could mean that people with greater short-term memory remember that blueberries are something that's very healthy to eat, and so those with greater short-term memory tend to eat more blueberries. Here's another example of a third variable. If you've been on a plane, you know that when the seatbelt icon lights up, you know that it's likely that there's going to be turbulence on the plane. And this little kid has observed that every time that light comes on, the plane starts to feel bumpy. So he's associated, just because those two things happen at the same point in time, they must be causal. But the truth is there's a third variable in this case, and the third variable is the fact that the plane is experiencing turbulence. That turbulence, or the threat of turbulence, causes the pilot to turn on the seatbelt sign, and it also causes the plane to experience that bumpiness. So two things that may appear to be correlated may not actually be, because there may be another variable that's actually causing them to appear that they're correlated when they're really not. Thinking about these different experimental methods, when is each appropriate? The important thing to remember is that as there's no perfect data source, there really is no perfect research design either. And what you find is that each of the designs has its strengths and its weaknesses, and often the weaknesses of one design actually are the strengths of another. So it benefits you to use more than one research design in investigating your research question. Also, the choice of your design depends somewhat on not only the question, but also the funding and what your goal is. Do you want to show that one variable causes another, or would you like to determine whether there's a relationship between two variables? And if there is, how strong of a relationship is that? The bottom line is that these different designs each provide complementary methods for exploring personality. The best idea is to conduct multiple studies using different experimental design so that you get more information about what you are researching. More information is like having more clues, and more clues gives you a better idea of the relationship between variables.